There are three types of specialization in plasma lamella. First is evagination, invagination, and junction complex. These are the three types. Outpushing evagination, which includes microvilli, flagella, cilia, and stereocilia. Inpushing, that is invagination, which consists of pores, mesosomes, lomasosomes, and transverse cell. If we see the junction complex, they are connected between the adjacent cells, and it have a width of 15 to 20 nanometers, which is filled with tissue fluid. There is a cementing material which is known as adrene, and the function as occludens, and there is a spot as mucula and a strip as zonula. Common junction complex are plasmodesmata, gap junction, intercellular bridges, tight junction, desmosome, and terminal bar. If we see a microvilli, there are numerous fine plasma lamella, which give straight and brush border appearance under the microscope. These area in between the microvilli are specialized for absorption. Surface area is increased in several times. For example, in the epithelial, intestine, hepatic cells, in the lining of the gallbladder and uterus. If we see stereocilia, a non-motile elongated evagination of plasma membrane, secretory or sensory. Example, macula, cyst, and epididymis. True cilia and flagella are covered by plasma membrane sheets. Evagination also occurs during formation of phagocytic vesicles. If we see a pore at a plasma membrane, it is connected with endoplasmic reticulum, and it forms a pore leading to a channel of ER, and there is an infolding which developed during the formation of endocytic vesicles. If we see a mesosome, it is a complex infolding of plasma membrane in the bacteria which connects the nucleoids, and it is believed to help that the nucleoid replicates, septum formation takes place, and even in respiration it is used. And there is an infolding found in fungi which is known as lomasosomes. Transverse cells, if we see a transverse cell, the cell wall as well as the plasma lamella, it shows infolding, and the cells are very specialized and it is used for the solute transport. If we see a plasmodesmata, they are cytoplasmic bridges between the plant cell and between the adjacent plant cell, which occur in a fine pore or pith in the cell wall. If we see a gap junction, there is a fine hydrophobic channel forming special protein cylinder of two adjacent cells. If we see intercellular bridges and interdigitations, the intercellular bridges consist of a plasma projections in the from their adjacent cell, which come in the contact with the intercellular spaces for the transfer of stimulus. If we see intercellular interdigitations, membrane outgrowths of adjacent cell, it fits into one another, it increases the adhesion and surface for exchange of the material. Then again next, desmosomes are present and tight junctions. If we see the tight junction, plasma lamella of two cells fuse to form a impermeable, example, epithelial cell or capillary or the brain cells. The function of tight junction differs in different tissues. If we see a terminal bar, the desmosome which lack tonofibril and where discoid thickening are replacing by the bands of microfibrils and intermediate filaments in the terminal bar. This was all about the specialization of plasma lamella.